Good. A very good afternoon here in Patancheru and good day any other place if other our colleagues are joining us. So today it's my pleasure to introduce or to have this PhD talk from Deepa Jaganathan, and she will be talking on fine mapping of QTL hotspot for drought tolerance in chickpea. And many of you already, well, so all of you who are here, so they already know that Deepa has been working with us as a PhD student for many years, and let me give the brief resume about her. So she completed her master's in biotechnology in 2007 at Bharathiyar University, Coimbatore. After that, she joined Indian Institute of Horticultural Research, Bangalore, and worked on various projects focusing marker generation and marker associated selection. Then she was a research associate with Rasi Seeds, Salem, okay. So then after that, she joined ICRISAT as a junior research fellow in ICRISAT DVT project, and she joined project in 2011 and that project, but then she moved to that new project, and this was genomics approaches for a stress tolerant chickpea as a senior research fellow. And when she moved to that project, she thought that she should do the PhD. Earlier, she did not have that plan. Her parents were saying that find a match. She says that I will complete my PhD. So eventually she decided that she needs to do that one. Is that right? Yeah. And then she opted for PhD and then joined LSU in 2013. At the same year, you were registered at Osmania University. And the PhD title is the same, fine mapping of QTL hotspot for drought tolerance in chickpea. And she completed her PhD work in the record time, so in just 3 years. So she started her PhD work in 2013 and then completed in 2016. Less than 3 years probably. So then during this period, she also visited Australian Centre for Plant Functional Genomics, Adelaide, South Australia for 2 months. And then although she completed her work, PhD work in just 2.5 years, but even in that during that time period, and because not only in the PhD work, but on the several other projects she was working. So she published 6 articles in various international reputed journals. And one of the articles called Genotyping-by Sequencing based intraspecific genetic map refines a QTL hotspot region for drought tolerance in chickpea got selected in the best 100 articles at International DNA Day conducted by Nature Groups of Springer. She also received Young Scientist Award from KV Rao Scientific Society, Hyderabad, obtained Best Poster Award in International Conference on Legume Genetics and Genomics and in 2012, and also obtained First Poster Award at Next Generation Genomics for Integrated Breeding and Crop Improvement Conference in 2015. So you can see that her career or her credentials are very remarkable as a PhD student, completing PhD very fast, publishing several papers, also having the international trip, and also won several awards. And so we are very much pleased about Deepa. Deepa's thesis has already been submitted, and I think that in next few weeks or so, we will be receiving, or she will be receiving the comments on her thesis to move ahead. But with these remarks, let's welcome Deepa for this talk. And I should also welcome our Deputy Director General Research, Dr. Peter Carvery, Dr. Purangor, and many other senior colleagues and all the friends and colleagues here. So with these things, Deepa, floor is yours. So please continue.
Thank you very much, sir. Good afternoon, one and all. I'm very happy to be here today because it's like a dream day. When every student were presenting, I was looking when it's my turn, and today is the day. And my PhD title is Fine Mapping of QTL Hotspot for Drought Tolerance in Chickpea. And in this presentation, I'll be covering introduction, objectives of my thesis work and results, summary and conclusion. So let's begin with the crop introduction. Chickpea, scientifically known as Cicer arietinum, and it's the second most important food legume crop globally, and it's originated from Turkey and Syria. It's a winter crop in semi-arid tropics and a spring or summer crop in Mediterranean and temperate regions. It's a self-pollinated crop and belongs to the family Fabaceae. Its diploid chromosome number is 2n is equal to 16 chromosomes, and its genome sequence has been recently decoded, like before in 2013, and the genome size of chickpea is 738 megabases. It includes around 28,000 genes. And chickpea is of two types, Daisy and Kabuli. Daisy, which is maroon in color, tough, everyone must be familiar of these types, and Kabuli, which is cream in color, having white color flower and soft coated seeds. So India is the major producer of chickpea and also major consumer. When we talk about production constraints, the abiotic stresses includes fusarium wilt, ascochyta blight, botrytis gray mold, and pod borer. When we talk about abiotic stress, drought comes as a major stress, followed by heat, cold, and salinity. However, among all the stress, drought plays an important role in yield loss of chickpea. So, and moreover, terminal drought stress is a major stress, as if we see in this graph, the climatic conditions of ICRISAT in October, which is the crop season for chickpea, and during its reproductive stage, like November to December, where the precipitation is very less and evaporation is more, the crop faces terminal drought, and which directly affects the yield. So in order to identify the genetic mechanism and improve drought tolerance, several studies were conducted at ICRISAT in breeding and physiology division, and two drought tolerance lines were identified. They are ICC 4958 and ICC 8261. They possess these characters like deeper root system, early maturing variety, and larger seed size. And whereas the sensitive lines are ICC 1882, ICC 283, and these lines possess shallow root system, late maturing variety, and they are smaller in seed size. Using these four genotypes, two different recombinant inbred lines were constructed. The first population includes ICC 4958 into ICC 1882. It comprises around 262 RILs, and the second population is ICC 283 into ICC 8261. This comprised of 281 RILs. And these populations were developed at breeding division ICRISAT, and these are all the root characteristics of the four genotypes. We can see a clear difference in ICC 4958 root and ICC 8261 root system with the sensitive lines ICC 1882 and ICC 283. And this is the seed characteristics of 4958 and 1882, and the seed size variation is very clear here. So the traits, in order to study the drought mechanism, extensive phenotyping was carried out under 5 different categories, right? Like root related traits, morphological traits, phenological traits, and yield related traits, and drought related traits. So let us see each traits in detail. So root traits, which included root length density, root dry weight, rooting depth, root surface area, root volume, and root ratio. So this work was conducted at ROS, rainout shelter, using these PVC pipes, and then the roots were washed, length were measured, and they were scanned for root volume and root surface area measurements. So in case of morphological traits, we had 5 traits, namely shoot dry weight, plant height, plant width, primary branches, and secondary branches. And phenological traits included days to 50% flowering, days to maturity. In case of yield and yield related traits, we had 6 traits, namely pods per plant, seeds per pod, 100 seed weight, biomass, harvest index, and yield. In case of transpiration efficiency, we measured, this work was carried out before I joined, delta carbon ratio and drought indices, drought tolerance indices, and drought susceptible indices. So using this extensive phenotypic data, genotypic data also was constructed on both the population, and the results are like in krill 3 is IC ICRISAT chickpea recombinant inbred line population, 03 is here ICC 4958 into 1882 population, whereas krill 4 is 283 into 8261 population. So krill 3 had around 241 loci map, which was spanning 621 centimargin, including SSR, EST, SSR, CAPS, and dot markers. Similarly, on krill 4 also, linkage map was constructed, which had 168 loci map, spanning 533 centimargin, including SSR, EST, SSR, CAPS, and dot markers. So QTL analysis was done using this phenotyping data and genotypic data. Around 76 major QTLs were obtained on krill3 and 51 QTLs were obtained on krill4. And when the QTL results were analyzed in detail, an interesting region on chromosome 4, which was having more than QTLs for more than 12 traits was identified, and the same region was identified on the other population also. So due to this importance of this region, it was called as QTL-hotspot. Just to make an importance, it is nothing related to recombination hotspot. Due to its importance, it is just called as a QTL-hotspot. And here is the map. This is a consensus map constructed by using this linkage group 4 of krill3 and krill4. So here we can see the QTL hotspot, and it has around some 5 or 6 or 7 SSR markers. Similarly here, very less number of markers, and it genetically spanned around 29 centimargan, and this was having around 700 genes and 7 megabase pairs on physical map. So this is a huge region. So due to its importance, we want to study more and identify what are all the genes present here, and we wanted to fine map this region. So this is the background of the work before I started my PhD work, and this was published in as a genetic dissection of drought tolerance in chickpea in TAG. So based on this background, my objectives was constructed as discovering more marker, that is SNP discovery and genotyping, and development of near isogenic line, and marker saturation in the QTL region, development of high resolution mapping population for fine mapping, and finally identification of candidate genes. Now I'll be presenting the results of each objectives. Objective 1, that is SNP discovery and genotyping. Here we followed Elche et al. Method. This is very popular genotyping by sequencing method. Here the DNA will be pooled and digested with the respective enzyme, which will be ligated with adapters, and the pooled samples will be PCR will be carried out for enriching the fragments, and the cleanup products will be used for evaluating the fragment size, and the selected fragments will be carried out for sequencing. And these sequenced samples will be looked for SNP calling, and finally SNPs will be generated. So using this approach, we have used our samples are ICC 4958, which is the tolerant parent, ICC 1882, which is the sensitive parent, and 208 reels. Due to some DNA check quality, we could not process all the RILs. We could get only 208 good quality DNA. So for all these lines, we carried out genotyping by sequencing, and the data generated here are present. Reads, totally 701 million reads were generated, which was around 59 GB on whole RIL, and the average coverage was 0.38x on RILs. So after SNP discovery, we got around 828 novel SNPs, and here is the summary of all the SNPs, how they are located all around the genome of chickpea. Here we got more number of SNPs on pseudomolecule 4, and less number of markers on pseudomolecule 5, that is 10. And using these marker data, genetic linkage map was constructed, and here just a snapshot of 3 linkage groups are given. Here this markers in red font are coming from GBS data, and these are coming from framework map, that is version 8 2014 paper, like we saw before in the background slides. So these are all 8 chromosomes, and when we see the summary, how many markers used, like these 828 are coming from this first objective, and all these SSR, EST SSR, GMM and dot markers are used from our earlier study. So totally 1146 markers were used, among which 1007 were able to map on 8 linkage groups. Around 88% of markers were able to map, and 139 markers were unlinked and not mapped. So this is the characteristics of linkage map, like it was having 1007 loci, and which includes 743 SNPs, 232 SSRs, 21 DART, 7 EST SSRs and 4 GMM. The map length was 727.29 centimargan. The highest number of markers were mapped on CALG4, that is 386 markers. Whereas, so therefore, highest density was also observed for linkage group 4, 3.44 per centimargan. Lowest marker density was observed for 7, that is 0.62 per centimargan. So using this data, we did QTL analysis. The phenotyping data used here was same as earlier used for preliminary QTL analysis, that is root, yield, morphological, phenological, physiological traits. And these data were recorded at 5 different locations in India and 5 different seasons. So similarly, along with the genotyping data generated newly and the phenotyping data of earlier, this is the summary of all the phenotyping data in detail. We did QTL analysis, and here are the results of QTL analysis. 164 major QTLs, that is QTLs possessing phenotypic variation more than 10% for 16 traits were obtained. And in detail, if we see for root traits, 8 QTLs were identified for 3 different traits: root length density, root surface area, root ratio. And for morphological traits, 34 QTLs were identified for 3 different traits, that is shoot dry height, plant height, primary branch. For phenological traits, we got 45 traits, and for days to flowering and days to maturity. For yield related trait, highest number of QTLs were observed, that is 73 for 100 seed weight, biomass, harvest index, pod and yield. Whereas for drought indexes, 4 QTLs were identified for drought tolerance and susceptibility index. So our aim is to check what happened in QTL hotspot, as we wanted to fine map this region. When we see QTL hotspot on linkage group 4, nearly 50% of the QTLs, like out of 164, 91 QTLs were located in this hotspot. Again, showing the importance of this region. And this region contained QTLs for 13 traits. The traits are mentioned here. And this region covered on physically nearly 30 megabase pairs. Earlier it was 7 MB, and now it is 3 MB containing 286 genes. So in summary, it was this is the status in primary analysis. We had such a big region, whereas after this objective 1, this inserting new SNP markers, this region has come up to 14 centimargan. Initially it was having around 5 or 6 SSRs. Now currently we have totally 55 markers here, out of which 49 are newly generated SNP markers. So this has brought down nearly half of the region into shorter, like 14 centimargan. So in order to use these newly inserted marker, as SNP cannot be used directly for any genotyping purpose, we were converted all the markers into CAPS or dCAPS. So for these sequences of these flanking sequences, around 60 base pair were taken and used dCAPS finder software, which given primers for CAPS and dCAPS, and we tried on all the parents initially for polymorphic checking. Around 16 CAPS, and out of 49, 16 CAPS and 33 dCAPS were generated, among which 14 markers were successfully converted into CAPS or dCAPS, 13 were CAPS and 1 was dCAPS. So here all the profile of 14 markers are given. Here are the 4 parents, like this is, sorry I didn't mention, this is 4958, this is 1882, 283, this is 8261. So in each slide we can see clear variation among the sensitive and tolerant parents. These can be used in any marker-assisted breeding program. So to check the candidate region from GBS approach, the flanking markers of QTL hotspot, like this marker and this one, the flanking markers were taken and aligned on chickpea genome, which covered, as we saw, around 2.83 MB region, having 286 genes. Gene ontology studies were conducted for these genes, and we could found some interesting stress-related genes like DREB, serine protein kinase, E3 ubiquitin, protein MIP transcription factor, heat stress transcription factor, all this. So still this region is huge, as 286 is a huge number of gene, and this 2 MB is not a fine map region. So still more markers needs to be generated, and This work was published in Molecular Genetics and Genomics last year. Then side by side, this development of near isogenic lines also we have done. This is for studying our QTL hotspot region in detail. Just to avoid the background of 1882, we brought the donor fragment from 4958 so that we will be evaluating our donor region by generating nil population. We will be checking our donor region's action on all the traits are in drought tolerance. So this is the scheme. 1882 sensitive line crossed with 4958 and F1s. After every cross with the sensitive line, foreground selection was carried out. Heterozygous plants were selected, and also background selection using the markers located on other region of other than donor region. Those markers were used for background selection, and for foreground selection, the marker located within the hotspot region were used. This was carried out at each generation until we obtained near isogenic line. So this is the summary of around like 3 to 4 years work. So here what we did, these are the foreground markers which were located in the QTL hotspot region, and for each generation, for example, backcross 1 F1, around 34 population was around 24, where we used two markers, ICCM 0249 and NCPGR21. Here 17 were heterozygous for this marker and 20 were heterozygous for NCPGR21. Then we chose the plants which are common heterozygotes, 14, and these 14 were forwarded for next generation to be crossed with 1882. So in this way, at every generation, foreground selection was done and it was carried to next generation. In backcross 3, we did background selection also. So here when we checked, common heterozygotes were 7. On these 7 markers, we did 35 markers which are coming from other linkage groups other than QTL hotspot region, and we could get around 80 to 100% of genome recovery. So then these plants which are having 80 to 100 were forwarded for the next generation, and similar work was conducted like foreground selection. And in backcross 5, we have used all the markers because here we wanted to, so we have used all the markers and common heterozygotes were 22. All the 22 were used for background analysis. Here we could obtain more, we could get more genome recovery, that is 92 to 100%. From here we have taken the plants which are showing 100% background recovery were carried out for fine mapping population development. They were termed as nil plus line, as their background is 100% like 1882 and they were carrying their donor segment. So for other purpose, evaluation and other field trials, this was carried out at breeding. And for our fine mapping purpose, we have used here nil plus line. So this is just snapshots for showing how foreground markers were developed. Here like the markers which are having, for example, this is Progeny, which was having both the fragments from each parent was selected. Excuse me. Similarly, this is the background marker profile, and for here the progeny which was looking like the sensitive parent was selected. So now we will be seeing the results of objective 3, marker saturation in the QTL region. So as we saw, the marker, the QTL hotspot was 3 MB and it was having 286 genes. It was a huge region, so we again carried out WGRS on this whole RIL population, that is krill3 ICC 4958 into 1882. Here is the data presented. On 4958 we got around 5.9 GB and 1882 5.94 GB, and on RIL 497 GB. So in total, while you analyzing this data, we could get around 62,000 SNPs, and these SNPs are like a huge number to be handled by any mapping software. So we here followed a approach which was already followed successfully in rice, that is bin map construction. So here just a brief introduction for what is bin and how we construct. Like this is parent 1, let us consider this is parent 2, and when we see the progeny, it will, like for example, if it had a recombination, it will look like this. And if we have for each of this locus a marker, let us imagine if we have marker for all these locus, then the genotyping data will look like this. So when we have this more than around 10 markers we are having, for example, the information what we get is there is a single recombination here. So instead of using all these markers, we can just tell it this is one fragment, this is one fragment. Call it as bin 1 and bin 2. So bin is nothing but the segment which is between two recombination breakpoint. So in this way, we are reducing the marker and also not losing our information. So this was, so the approach is called sliding window approach. It was presented in detail in Hong et al. I'll be just telling the overall approach. So here what happens, like let us consider this is one chromosome and each color is like one particular parent type. Red or like 4958, blue or 1882 for example. So here each, 15 base pair window will move throughout the chromosome for 100 kb region. So at each 15 base pair, and this is the table to present the genotype. Like for example, if 15A is found and 0B is found, then it will be represented as, this is for rice, so it will be represented as A type. Same way up to 11 to 4. Like this, this window keep on moving and this will be represented like whether it is A genotype, whether it is B genotype, or whether it is heterozygote. So this places heterozygote here when it is continuously accounting like 8 to 7, 7 to 8, 6 to 9, this type of AB, if it is getting, it places heterozygote. So from this, like this kind of SNP information is coded like this, and we are getting like these bins. So this much marker information is just coded as 4. So this is the way bin map is constructed. So now this much information is converted like this. So this is on for one RIL, for whole population, all the RILs were constructed like this, and overall for all the chromosomes will be coded and total bins will be obtained. So here is the results of our work. The number of SNP used here was around 53,000 and 54 SNPs which are coming from our objective 1. And as said, that sliding window approach was followed. Number of bins here obtained was out of 50,000 markers, 1,610 bins we obtained, and more than 60% of the bins were having less than or equal to 1 megabase per size. So here we can imagine like more number of recombinations were covered in this study. Like 60% of the bins are less than 1 MB, and 70% of the bins were having less than or equal to 10 genes. So, and average number of bins per RIL were 36. Average number of bins per chromosome was 201, which varied from 112 bin to 281 bin. More number of bins, as our aim is on QTL-hotspot, now I am presenting number of bins on CALG4 281, and inside QTL-hotspot we got 73 bins. So this is overall summary how the recombination breakpoints were looking on chickpea whole genome. And this is our chromosome length, and this is number of bins we obtained, and this is the minimum bin size that is obtained for chromosome 4, 41, and the maximum bin size was 8 on linkage group 7. So this is the distribution of bin size. As we saw, like highest percentage of bins were less than 0.5 or up to 0.5 megabase pairs, whereas around 70% of the genes were having, bins were having less than 10 genes. So using this 1,610 bins as markers, we constructed linkage map, and this is the summary of the linkage map, and it was having around 1,557 markers, which spanned about 973.94 centimargan. This picture depicts the overall recombination patterns. All the bins represented, each line is one RIL, and here green color is 4958 and red color is 1882, and this shows the complete genomic pattern, recombination pattern on chromosomes. So using this map, QTL analysis was conducted and 134 QTLs were identified, 71 were major QTLs. So this is the results of QTL. So when we got 134 QTLs, for example, for days to maturity, we got, for example, if we got 10 QTLs, we have just checked which is having highest phenotypic variation. In such way, in this table, only the QTLs are represented which are having highest phenotyping variation. So for days to maturity, we got on LG7, and for within QTL-hotspot, we found one cluster where we found QTLs for 5 traits. Excuse me. And the other cluster, which was having 2 traits, and on chromosome 8 we got for 3 traits. This is the summary. So this was preliminary analysis where we had only 6 SSR markers. This is from objective 1 where we have 55 newly generated SNPs, and this is the current bin mapping study. This is the cluster 1, cluster 2. We now name it as QTL-hotspot A and QTL-hotspot B. QTL-hotspot A has QTLs for 5 traits and QTL-hotspot D has QTLs for 2 traits. And this is the J-browse view for how many genes are located in each QTL-hotspot. Like QTL-hotspot A is of 139 kb size and it is having 15 genes. QTL-hotspot B is of 153 kb size. It is having 11 genes. This was published in scientific reports. Next we will be seeing about development of high resolution population. Here near isogenic line coming from objective 2, which we termed already NIL plus line, which was mapped with sensitive line 1882, and the idea is to produce F1s and generate a large number of F2, which will allow more segregation and more recombination in our QTL-hotspot A and B, so that we can validate that regions and also bring them into pinpoint into single gene level. This is our aim, and we have constructed this high resolution mapping population. And on F2 we have done marker analysis for selecting the recombinants, and in F4 we want to fix the recombinants which are having heterozygous allele, and in F4 we were doing progeny testing and candidate gene identification. This is overall flow. Now we will see in detail the results. These are all F1, F2, F3 populations at different locations in Patancheru and Dharwad. And so here, in order to screen the large mapping population F2, we have taken all the SNPs located within QTL-hotspot A and B, and the flanking markers and one marker from middle of the region. This is QTL-hotspot A, QTL-hotspot B. Flanking markers and one from middle were taken and converted into KASPar. KASPar markers are just allele specific genotyping marker system. And here snapshots of all the 6 flanking markers and 4 flanking markers, 1 middle marker. Here each dot represents one particular plant, and like red or homozygous for 495 allele, green spots are heterozygous allele, blue spots are homozygous for 182 allele. In this way, we have phenotyped around 2000 plants. All the 6 markers were genotyped here. The parental types are 50% we observed parental type, 50% we observed the heterozygous type, and we selected heterozygous recombinants which were of 42. Though it was 980, we selected only 42 because we didn't choose the marker which are having all H, because it will be tough for us in the next generation to fix the alleles. Therefore, we chose only 42 best lines where already some alleles are fixed, like B, A. Due to less seed number, we have only forwarded 19 plants. And this is the F3 data, where from 19 plants, 284 plants were generated. In this parental type were 159, heterozygotes were 125. Though we got 125 heterozygous recombinants, we selected only homozygous recombinants. They are represented here. Here it shows recombination happened here and the alleles are fixed. And each types we have here now homozygous recombinants on which we carried out phenotyping. So this is the summary. Around 2000 F2 plants were analyzed, 19 were forwarded, and these 19 families gave us 284 progenies. And on this 284 progenies, 59 F3 homozygous recombinant lines from 10 out of 19 families were used for phenotyping. So this is the result of phenotyping. For this F3 homozygous line screening, we used all the KASPar markers. Phenotyping data was constructed only for 100 seed weight. Due to certain unfavorable condition, we could not measure for all the traits. And these are all the 10 families which are depicting the same genomic constitution. And if we see the progeny testing, like this is the 100 seed weight, and we can see this fragment QTL-hotspot A region directly influencing the yield. So when we get this red are 4958 allele, blue colors segments are 1882 alleles. So we can see the clear-cut difference that 4958 alleles are giving more yield. So this is and then progeny testing also was confirmed by QTL analysis. This is our QTL-hotspot A and B. This is the map generated using 6 flanking markers on F2. This is the map generated on F3 using all the KASPar markers. And when we conducted QTL analysis on F3 homozygous lines, this was clearly placed on QTL-hotspot A, and which was having around 15 genes. So now we will be seeing the results of last objective, identification of candidate genes. And here we represent QTL-hotspot A, like all the 15 genes were represented. More transcription factors were found, and some stress-related genes like serine threonine protein and early light-inducing proteins were found. And this is from QTL-hotspot B. Still we are considering this region because we have not yet screened for all other traits. And here also important genes were found. And in summary, objective 1 and 2 summary will be like ICC 4958, we generated 6.24 GB, and on 1882 5.65 GB. The RIL data had 59.03 GB. Totally 828 novel markers were developed, and it helped us to generate high density map of 1007 loci spanning 727.29 centimargin. Using this map, the QTLs were analyzed. 164 QTLs were obtained, and 49 SNP markers were located in the QTL-hotspot region, which brought down into 14 centimargin. 3 MB it was covering on physical map, having 286 genes, and new markers like 14 caps and 1 dCAPS were generated. This can be used in breeding programs, and near isogenic line were for validation and fine mapping population development were developed. This is the result of objective 3. Around 53,000 SNPs were generated, which gave us around 1610 recombination bin, and the linkage map size was now 973.94 with 1500 bins. The QTL study revealed 71 major QTLs, 63 minor QTLs. Major QTLs were obtained for 11 traits, whereas minor QTLs were obtained for all the traits. The QTL-hotspot region was now refined into QTL-hotspot A and B. A is of 139 kb and B is of 153 kb. 15 genes were obtained in QTL-hotspot A, 11 genes were obtained in QTL-hotspot B. The result summary of objective 4, high resolution mapping population was constructed by crossing NIL+1 line into 1882. Around 6,000 F2 plants were generated, out of which 2,000 lines were used for selecting the recombinant lines. KASPar assays were developed for all the SNPs located in 2 QTL-hotspot regions. Genotypic analysis of F1 lines revealed 42 recombinants. Progeny testing on the selected recombinants in F3 lines revealed QTL-hotspot A in controlling 100 seed weight. This is the summary of objective 5. QTL-hotspot region refined into 2 sub-regions, and these are all the important genes found in those regions. The final outcomes is large number of markers were generated. Genic markers also were generated to be used in breeding program. High density linkage map is available now. Fine mapping population was developed for validating the refined QTL regions. QTL-hotspot is delimited into A and B. Novel candidate genes were identified. The future direction can be these, for all the traits we can do the progeny testing on F4 population, and once we reach a very less number of gene level, detailed analysis can be carried out like gene expression, complement testing, knockdown experiments, proteomic analysis also, and finally it can lead to QTL cloning. So this work, first two these are all the publications I got from this work. First two are directly coming from PhD work, and all other four are from related works. And these are all the abstracts presented at various international conferences. And I would like to acknowledge my supervisor, Dr. Rajiv Varshni, for giving me this opportunity and having the confidence that I can handle such a huge project. Thank you, sir. And Dr. Puran Gaur, for like I always run with problems. I have less number of seeds, plants are not grown. So he always guide like fatherly guidance. Thank you, sir. And Dr. Mahendra Turi, thank you, sir, for guiding me, like especially in paper writing and all this stuff throughout the study. And Dr. L. Krishnamurthy, he is not here, but he helped me a lot in learning physiological aspects. And all other friends and colleagues, Dr. Manish Rukibal, Dr. Sandeep, Dr. Srinivasan Swamini, and each and every one of the members in CEG, everybody's contribution is here in the form, and library, FETS, housing, and other groups, ACPFG, Dr. Tim Sutton from UWA, Dr. Dave Edwards, Dr. Jacqueline Bartley, and Mr. Pradeep Ruparu from Usmania, my co-supervisor, Dr. Kavikishwar, and other teaching and non-teaching staff. Thank you.
Thank you very much, Deepa, for very good presentation and highlighting several important activities from her work. Take water. So yeah. So if you have some question, clarification from Deepa, I hope that she will be very happy to mention. And good thing is that you already acknowledged contributions several of senior people from here on the different slides, especially from Dr. Puran Gaur, Dr. Krishnamurthy, who played very critical role in this thing. So this was really good. And Srinivasan also played important role, and Mahendra, so this was good. Manish is working in last. Anyway, so I can see that there are many friends of Deepa, so maybe they will not ask the question, but this is, if you will not ask the question, you will not be offered a cup of tea or coffee, because after that we will be going there. So it's good to ask some question to Deepa, to have some explanation, because I know that our DTG is already having questions. So before he asks question, some of you should ask the question. Okay, please go ahead, Manish. Yeah.
Deepa, it was like a very nice work and nice presentation. Maybe you finished your bottle while presenting drought tolerance QTL. My question is, you got, apart from drought tolerance, you got good QTLs for other traits as well, like seed weight and many other associated traits like harvest index, I think.
Yeah, for
so, like, yes. So, have you arrived to some markers that they are genes for those traits as well?
Yeah, actually, this region is containing lot of transcription factors. What I think or we think is like it is not directly gene. We cannot directly link to any one gene or something. There must be some like promoter or some region controlling for genes of all other traits, genes which are responsible for each trait. Those controlling region must be this.
Okay. Any other question? Yes, sir. Peter, here, please.
Deepa, thanks very much for the presentation, and congratulations on getting to this point. It's always a relief when you finish your PhD. And I hope by the time I leave ICRISAT I'm going to understand more and more of these seminars. It's not an area that I well know. But my question to you is, as a consequence of your work, what is Puran Gaur going to do differently next season?
Apart from this,
of your work, the work you've done and the markers you've found, will Puran do things differently in his breeding program? Can you Can you tell me what he's going to do differently?
So the near isogenic line program is completely coming from breeding. Like they're already doing validation at different fields. So that is going in that direction. And in fine mapping population, we can move ahead for next generation on F4, studying the rest of the traits.
So I know that Peter will or hates me for explaining the question back to Deepa. His question is that whether your work has provided something that can be used by breeders for the drought tolerance related stuff.
Yes.
Yeah, so you need to answer that one, not that one, what Dr. Gore is doing.
No, what he will be doing here.
So that like QTL-hotspot markers, whether he can use, so in those direction he needs to. So you need to answer in that direction. So can you answer this question again? Sorry about that, but.
Sorry, I didn't get the question.
So answer that one now, yeah.
So yeah, actually we saw here those markers directly can be used for
And are
selection.
being used. And are being used.
Yes, and are being used. And already this donor segment has been in progress in different lines in African locations, and other programs they have taken up. And these markers are already used in by various other groups also, like in NIPGR, these markers.
IRI, ILPR, many institutes.
Yes.
So you see, Peter, that you need sometimes people like me who can explain that what is the question. Anyway, any other question? Kartavya, please.
Excellent work, Deepa. Just this QTL region, when we initially said 91 QTLs, 286 genes, and then when you have two sections, A and B, then sometimes you said 15 genes and 11 genes. So how these relate actually? You say one region, 15 gene, 11 region, but when you say together 286 genes, when you say candidate genes, then 26 genes. So how do we relate these? Can you give something on this?
Yes, sir. Sir, like before fine mapping, we were not knowing. So we were calling like initially 700 genes we were holding, then 286 genes we were holding. Though even there we are not telling all of those 700 or 286 are important. So to bring out what are all the important genes, we did fine mapping, and finally we are ending up with 26 genes. So we consider these are now candidate genes. If we work on further, we can directly pinpoint like very few genes, 5, 6.
And to add that one, we should say that these 26 genes, they are now in those peaks where we have the QTLs, but still this cannot be told that, well, so all these 26 genes are like that one, so you need to have much more analysis to reach the point, and even not with the QTL hotspot or drought tolerance is very complex thing, so rather the traits which you are talking and the QTLs in those peaks you are having, that this particular gene may be associated with that kind of thing. So again, majority of time in this genomics research and this informatics, you have the association, not the causality. Causality will come later, that once you reach at that stage. But then for breeding application, association enough. Association is good enough to use them in the breeding program, because in the genome you are having the big LD block. So even if you are a little bit far from the gene, this will be helpful in the breeding. Okay, any other question?
Deepa, one more question. So in this QTL region, like you have some traits, so you have like conflict between those genes as well. Suppose for one desirable trait you have negative effect or something like that. Because did you show that?
No, not checked.
So suppose if you interrogate this region and you are getting some undesirable
I
traits.
got your question, but I don't think we have not checked.
Okay.
And then you also have the truth. Please.
Deepa, one more question. You looked at root length density and rooting depth. Did you partition your root length density up into layers so you can see whether the concentration of roots was in the surface or at depth? Because for drought tolerance, that's an important, total root length is important, but also its distribution down the profile. You didn't, and so we don't know or whether your markers are relating to root length at depth or concentration in the surface. But you must have the data to be able to look at that.
Dr. Gaurya, please use mic so that people can-
The density was worked out. Yeah, it's there. What is in first 15 and then 15 and like that. Yeah. Okay. Because she didn't see that is done by the earlier. She used the data.
So use the data, but this analysis, yeah, so
Was done.
you have the,
Yeah, and correlation is there, but we have not studied in fine mapping population. In like, if you refer dissection paper, in that the correlations are given for like yield and root length. Yeah.
those were already established studies long back when they started that roots stuff. So Junuchi, Kashi, Bagi, Krishnamurti, that what kind of correlation you have, and they were well established study, but see did not do those kind of moderate drought and this thing. So this was it. So Mahendra, you had some question or no? Okay. Any other question? So if there is no other question, then let's thank once again Deepa and congratulations to Deepa. So Deepa is still working on that, I think last paper, so together with Sandeep Kale, so she will be here for few more months probably or like that, because she is also waiting for that PhD completion. Well, you submitted, but she will do this thing, so you will keep on seeing her.